OK. Ben, à cause du temps, on va continuer. Puis je pense qu'il y a, de toute façon, on va avoir un peu de temps après pour discuter. Puis on a aussi notre séance de brainstorming entre chercheurs du réseau qui va être après. Donc maintenant, ça me fait plaisir de vous présenter la dernière conférencière de notre. Attends un petit peu. Ça se peut que tu peux débrancher. La dernière conférencière de notre belle journée, c'est Dr. Sarah Kimmins. Alors, je recherche mon petit papier. Sarah est professeure agrégée en biologie de la reproduction à l'Université McGill. Elle est titulaire d'une chaire de recherche du Canada en épigénétique, reproduction et développement. Elle est largement supportée par les organismes subventionnaires de haut niveau. Elle a une expertise qui est tout à fait complémentaire avec celle qu'on a entendue jusqu'à maintenant, où en fait elle s'intéresse à la fertilité du papa, la fertilité de l'homme et comment l'environnement paternel a un impact sur la spermatogénèse et donc le développement embryonnaire et la santé de l'enfant. Donc elle nous rappelle que ce n'est pas juste mère-enfant, mais que c'est parent-enfant, ce qu'on est en train d'étudier, et c'est extrêmement important. Donc, Sarah va nous parler de l'épigénome du sperme qui influe sur le développement embryonnaire et la transmission de maladies de façon intergénérationnelle. Sa présentation va être en anglais, mais je suis sûre qu'il n'y aura pas de souci pour qu'on pose les questions en français, même si elle répond en anglais. Voilà, donc Sarah, à toi, merci beaucoup d'être là.
Merci. Et merci à tous les présenteurs avant moi, especially Marc-André, for doing such a nice job of introducing epigenetics. And that'll be very important. And I can skip over some of mine, and maybe we'll have some time for questions at the end, even though it's Friday. So the interconnection between all humans and the environment lies within the epigenome. Yet we don't really understand how these environment epigenetic interactions lead to diseases such as diabetes, neurological disease, and cancer. And so our research group is studying how the environment in particular influences the sperm epigenome, which is heritable, and how that can lead to disease in the next generation. And so today I'm going to present to you some of our work from mouse models. And then also in particular, one study where we've looked at the exposure of men living in Greenland and South Africa to DDT and how this has changed their sperm epigenome and how that relates to population health in those areas. So our understanding of the heritability of information through DNA has changed drastically in the last 30 years. We used to think it was solely based on the DNA sequence, and now we know that inheritance also includes this epigenetic layer of information, which takes the form of DNA methylation localized to CpGs, as well as non-coding RNA and modifications to histones. So we currently define epigenetic inheritance as non-DNA sequence-based inheritance that changes gene function from one generation to the next. So the first example of how environment and health can interact transgenerationally came from this study that was published not too long ago in 2007 by Gunter Kati and Marcus Pembry and their colleagues. And they studied the food records and health records of a very unique area called Överkalix, which is located in Sweden. You can see it here in the map. And it's a very small community, and it stood out because they kept such meticulous records, birth, death records, records of disease, and records of food availability. And so they looked at 20 years of good food availability and 21 years of poor food availability. And they assessed this access to food during puberty in 3 cohorts in 1890, 1905, and 1920. And what they found was that when the father or grandfather experienced famine, his children tended to be protected from cardiovascular disease and diabetes. In contrast, when there was food abundance during the slow growth period just before puberty. This was associated with a shortened lifespan that was associated with either diabetes or heart disease. And so this was the first epidemiological study to demonstrate transgenerational inheritance. And importantly, it suggested that the puberty period, which coincides with the initiation of spermatogenesis, may be a particularly sensitive time in life for nutritional programming of developing sperm. So we study what we call the field of POHAD, which is paternal origins of adult health and disease. And shown here is a schematic which summarizes our working hypothesis of most of our studies, including the ones I'll present today. So we believe that paternal exposures, things like exposures to stress, diet, or toxicants, can alter fertility. And at the same time, they potentially alter the sperm epigenome. These changes in the sperm epigenome are then transmitted at fertilization. They persist in the developing embryo, resulting in subsequent changes in gene expression, which can impact fetal development, placental development, for example. And then later, when that fetus grows through childhood and adulthood, this could lead to a predisposition to complex diseases and altered phenotypes. So I won't spend too much time here, as Marc-André explained very nicely the mechanisms of epigenetic inheritance. The ones I'll focus on today involve DNA methylation and modification to histones. In particular, we look at a gene activating histone, histone H3, lysine 4, which is tri or dimethylated. And that's heavily found at things like gene promoters, for example. For us, I think what's important to highlight here is where are the gaps in knowledge, and what do we define as a mark being involved in epigenetic inheritance. So for us to demonstrate that an epigenetic mark is responsive to environment and could be involved in epigenetic inheritance, we've wanted to show that this mark can persist in the germline, that it's transmitted at fertilization, which means tracking it through to the embryo, and show that it's able to escape reprogramming so that it can provide a cellular memory in subsequent generations. And it's been really difficult to determine and discover evidence of such an epigenetic factor that meets this criteria. We have been able to do it in animal models in some situations, but doing this in men has been more challenging for obvious reasons. One, for example, being that it's very difficult to get human embryos to work with, and that makes it hard to track the changes in the sperm directly to the embryo. For those of you that aren't very familiar with the sperm, I'm going to give a little bit of background here about the very unique sperm epigenome. So sperm, unlike any somatic cell, is designed to leave the body and enter another body and deliver its epigenetic and genetic material. And so it has an incredibly unique design to allow for that function. And this includes having the sperm tail and also the very unique organization of the genetic and epigenetic material. So unlike a somatic cell, the histones in the sperm are predominantly evicted during spermiogenesis. So very few histones are retained. In the mouse, about 1% of histones are retained, and in men, about 10% of histones are retained. And where those are retained, we used to think that that might just be random and was a consequence of incomplete eviction of the histones. But in some later studies, we were actually able to show that the histones and where they're retained serve a very important role in terms of gene expression in spermatogenesis and in embryogenesis. So our research group, along with others, has shown that there are two programs, epigenetic programs, that are marked in sperm, and that meaning that you can track back where you find these histones, for example, to genes that are involved in regulating fertility and spermatogenesis. And then also there's a future program that's mapped in the sperm on the epigenome. And this is a program that's laid out on the genes that will be expressed early in embryo development. So we've been using mouse models to identify epigenetic modifications that could be implicated in epigenetic inheritance through the paternal germline. And the types of questions we're asking are what epigenetic modifications can escape reprogramming during spermatogenesis and embryogenesis? What epigenetic modifications are sensitive to the environment? And today I'm going to tell you about one of our dietary models, a folate deficient model. And we've also been asking whether these environmentally induced epigenetic mutations can be retained in the embryo, and are they associated with altered gene expression in the embryo, and then later disease phenotypes in the adults. So this was a foundational study for our work going forward. It was published a number of years ago. And as I mentioned, when the field first started considering the role of histones in sperm, it really wasn't clear whether histones were actually important in sperm and doing anything in terms of governing gene expression in the next generation. So we wanted to answer that question, and to do so, we designed a transgenic mouse model. And what we did, you can see the construct here, is we overexpressed a histone demethylase known as KDM1, lysine-specific demethylase 1. And this is an enzyme that removes methyl groups on histone H3, lysine 4, in particular mono and dimethylation. And this is a histone mark that you tend to find at gene promoters, for example. So we designed these transgenic mice, and the first thing we did is we bred the transgenic males, which we keep as heterozygotes, to wild-type females. And to our surprise, what we found is that we had very severe birth defects. Some of these examples you can see up here. We had major problems in development, including mostly changes in patterning. So we had things like missing limbs, missing eyes, underdevelopment, runting. There were also problems in things like kidney function, and also some behavioral changes. So we wanted to know, where in the sperm did we change methylation? And so to do that, we used a technique called chromatin immunoprecipitation, and we followed that by sequencing. And the results of that are shown here on these heat maps. And I just want to draw your attention to the lanes with H3K4 dimethylation. So where you have the bright spots down the lanes is where you have the mark of interest that's targeted by KDM1. And you can see you have levels of that mark all the way down in the wild type sperm, whereas in the transgenic sperm, you can see there's this deep trough where you have missing H3K4 dimethylation enrichment. And when we looked at what genes were there, those are genes that are involved, for example, in embryo development and spermatogenesis. And many of those genes corresponded to genes that are deregulated in expression in the embryos that are sired by these transgenics. When we kept breeding these animals for subsequent generations, we found that there was a transgenerational phenotype. So even when we bred non-transgenics, so mice that had a transgenic father but didn't carry the transgene themselves, their offspring were abnormal. And so we knew that we had a very powerful model to study transgenerational inheritance. But what we didn't know is what mark was actually driving that transgenerational inheritance. So when we looked, and I'm going to draw your attention to the column here on the heat map, when we looked at H3K4 dimethylation in the non-transgenic sperm, we found that it actually wasn't altered. So this told us that we have a transgenerational phenotype, but it's not being driven by H3K4 dimethylation. And so to answer that question, we carried on with a new study with this mouse model, the results of which are shown here. And basically, to summarize, what we found is when we profiled H3K4 trimethylation in the sperm from the non-transgenics, we found that that mark was altered at regions that correspond as well to the phenotypes that we observed. So H3K4 trimethylation, although it's not a direct target of KDM1, what we had done is we had changed the balance of activity of histone methylases and histone demethylases, and that resulted in a consequent increase in H3K4 trimethylation enrichment in the sperm of transgenics that was able to escape reprogramming and persisted in the sperm of the non-transgenics. So that told us that we now have a mark that we can study that we know is involved in transgenerational inheritance, at least in the mouse model. The next step was to determine whether our mark of interest, H3K4 trimethylation, is actually sensitive to environmental stressors. And so now I'm going to present the work of a talented PhD student in the lab, Ariane Lismer. And so she wanted to answer this question of whether the sperm would be responsive to an environmental challenge. And in this case, she used a diet that was folate deficient. So why folate? Well, we are interested in folate for several reasons. The first reason being that 1-carbon metabolism feeds directly into DNA methylation and histone methylation and protein methylation. So if you change 1-carbon metabolism by changing the availability of folate, you change essentially the methyl donor availability, and you can change the epigenome. So we knew it was a direct route and exposure to change the epigenome in developing sperm. The second reason we were interested in using this diet is for its translational interest. Even though there's folic acid food fortification in as many, I think, as 70-something countries worldwide, there still are populations that are folate deficient, including in Canada's north, where you have households that are food insecure. So folate deficiency is not only impacted by diet, you can also have folate deficiency induced by alcohol consumption, smoking, and it can be influenced by high levels of homocysteine, which can occur, for example, in people that are overweight or have a severely elevated BMI. So here we have a schematic showing the mouse model that Ariane used in her studies. She used wild-type mice. These males were either fed a folate sufficient or a folate deficient. And then she also used the transgenic model, so the mice that overexpress KDM1. And the reason she used that mouse model is she wanted to ask the question experimentally of whether there could be cumulative effects of epigenetic stress on the sperm. Meaning so that if you have a predisposition and an exposure that has already altered the sperm epigenome and you challenge that animal with another environmental stressor, in this case the diet, does that worsen the effect on the sperm epigenome, and what happens in terms of the offspring outcomes? And this could be relevant to human studies where, for example, you have a man who's been exposed in utero to folate deficiency, for example, or another environmental challenge, and then goes on through his life to be exposed to further stressors. It answers the question of, could that man, for example, be at a higher risk to have children with developmental abnormalities? So she bred these males. They stayed on the diet for several spermatogenic cycles, and then she looked at the pregnancy outcomes by looking at animals at embryonic day 18.5, and she performed a skeletal analysis as one of the readouts. So shown here are some of the results from that study. So here are fetal skeletons that have been stained for bone, which is shown in red, and cartilage, which is shown in blue. So we're looking at an offspring sired by a folate sufficient control here. And then we're looking at offspring that have been sired by the folate deficient transgenic. And you can see that there were severe abnormalities, craniofacial abnormalities in the offspring that were sired by the transgenics. And this was also occurred in the offspring that were sired by the wild type animals that were folate deficient, although the severe abnormalities were more persistent and occurred more in the transgenic offspring. So we had things like hypoossified skulls and abnormal craniofacial developments. We also had problems with vertebrae formation, problems with ribs, and the full data is shown in the paper if you're interested. But what stood out for us was that the total number of skeletal abnormalities was significantly higher in animals sired by the folate deficient transgenic than animals sired by the folate deficient wild type. And there were more severe abnormalities. So this indicates to us that you can have cumulative effects on the sperm epigenome that result in cumulative abnormalities and more severe abnormalities in the offspring. So the next step in the study was to look at the sperm epigenome to determine whether H3K4 trimethylation in the sperm was indeed sensitive and could be a readout for this environmental exposure. So to do that, Ariane performed chromatin immune precipitation followed by sequencing on sperm from wild type and transgenic animals. And here we're just looking at the data here from the wild type sperm. And what she found was that her differential analysis, looking for differences in enrichment in H3K4 trimethylation in the sperm, showed that there were both regions that gained and lost H3K4 trimethylation. And these occurred at different regions of the genome. So here's her genome ontology analysis shown here on these graphs. And you can see that regions that gained H3K4 trimethylation were predominantly found at transcriptional start sites close to promoters, and also throughout the genome at regions including intergenic regions. And this was also the case for regions that lost. She then wanted to gain some functional insight into what processes could be impacted by the changes in H3K4 enrichment. And what she found is that there was an enrichment for genes that are involved in post-embryonic development, early embryo development, regulation of bone remodeling, which feeds directly into the phenotype that she'd observed, as well as, for example, this osteoclast proliferation. So at this point, we're able to tie the changes in folate deficiency to changes in the sperm epigenome, to changes at genes that are involved in the phenotypes. The next question was to ask, could H3K4 trimethylation changes in the sperm be transmitted at fertilization, retained in the embryo, and lead to altered embryonic gene expression? If you remember, that's the basic criteria that I defined at the beginning of the talk as being required to be fulfilled for a mark to be evolved in epigenetic inheritance. So to answer those questions, she did several types of analysis. She performed chromatin immunoprecipitation on embryos that were sired by folate deficient fathers, and she also performed RNA sequencing. And I'll just draw your attention here to these heat maps first. So here what she's looking at is H3K4 trimethylation, where it's present in all the regions in the sperm. And you can see by this very bright band, here is H3K4 trimethylation in the sperm. If we look at where you find this mark in oocytes, you can see that it's the direct opposite of what we find in the sperm. So this suggests that if you look here at where you find H3K4 trimethylation in the 2-cell embryo, that this mark is coming from the paternal chromatin and is being driven by the sperm H3K4 trimethylation, not the oocyte early in development. And then by the 8-cell, you can see that this mark is still persisting. So here we're looking at these scatter plots where we're comparing H3K4 trimethylation in the 8-cell embryo at promoters to H3K4 trimethylation in the sperm, and the color indicates the level of gene expression. And what you can see here is that where you have H3K4 trimethylation in the sperm corresponds to where you find it in the embryo, in the 8-cell embryo, and you find this mark at regions where you have high gene expression. On the next scatter plot over here, what we're looking at is the genes that show differential, the promoters actually that show differential enrichment for H3K4 trimethylation. And you can see indeed that those genes have H3K4 trimethylation that's altered in the embryo by diet that persists and is associated with the altered gene expression in the folate deficient sired embryos. So as much as 25% of the promoters that have altered H3K4 trimethylation in the folate deficient wild type sperm and embryos show deregulated gene expression. So to summarize, we've shown that we've found a mark that is sensitive to the environment, in this case, folate deficiency. We've shown that paternal folate deficiency leads to birth defects that are observable in our model, and that you can have an accumulation of damage done to the sperm epigenome that can lead to a worsening of phenotype. And we've been able to identify a mark that's involved in epigenetic inheritance that is transmitted in the sperm, persists in the embryo, associated with abnormal gene expression and with the functional phenotypes. So the next thing to do was to see whether the same mark, H3K4 trimethylation, is also responsive to environment in men. So when we began this work, There was some data available on H3K4 trimethylation in sperm, but it was contradictory. And since those studies have been done, the methods for sequencing have greatly improved. And we've also optimized this method specifically for sperm. So what we wanted to do was generate a really deep sequencing reference population set of data showing where H3K4 trimethylation was in sperm from men. So we used what we call a reference population of 30 men, and we performed deep sequencing, and we had a very high number of reads using this pooled sample. We had 153 million reads. And what that allowed us to do was to identify where this mark was in sperm from men in regions that had not previously been identified as being enriched for this mark. So previously it had been shown that this mark was present at promoters, but with our deep sequencing, we were able to show that it's also present at other functional regions in the genome, including enhancers, intergenic regions, and also at transposable elements, which are incredibly interesting because they've been implicated in epigenetic inheritance, and they're also expressed very early in gene expression, so very early in the embryo in both mice and men. So in the same study, we also looked at where H3K4 trimethylation is in relation to DNA methylation. And what had previously been reported was that where you tended to find H3K4 trimethylation, this was mutually exclusive of where you find DNA methylation. And we found that that's not always the case, that you do have regions where the two marks can interact. And that's important going forward when we look at the sperm epigenome in response to the environment, to know that there can be interaction between these two epigenetic marks that are involved in inheritance. So the next thing we wanted to know is H3K4 trimethylation in men, like in the mice, responsive to environment? Can we use it as a readout of environmental exposures? And can it be associated with health outcomes in populations? So to do so, we asked the question of whether does exposure to the endocrine disruptor, DDT, impact DNA methylation and H3K4 trimethylation in sperm from men? And does this relate to poor child health in regions where there are high levels of DDT exposure? So despite being banned in the 1970s, DDT is still widely used. It's a persistent organic pollutant. It has estrogenic properties, and its metabolite, DDE, is a very potent anti-androgen. And so even though it's been banned, it still persists in the environment. And it's also important from the perspective to revisit the links between DDT and human health, because there has been discussion about increasing its use with the spread of the Zika virus. So DDT in particular is still used in India as well as in South Africa, and that's one of the regions for our study population. But it also it persists and travels to regions in the northern parts of the world, and this is due to things like ocean currents and different weather patterns. So Arctic populations, for example, are also exposed to DDT through its bioaccumulation in the food chain. So we studied two populations of men. We studied Greenlandic Inuit, as well as men who are living in the Vemby area, which is in the province of Limpopo in South Africa. And these men are Venda tribesmen, and they live in these traditional huts, as you see here, these mud huts. And it's an endemic malaria area. And so because of that, there's a government-controlled spraying. So they spray the inside of these huts in the villages where there's particularly high areas of malaria with DDT. So these men are directly exposed to some of the highest levels in the world. So regions where there are high levels of exposure to DDT have been associated with high levels of miscarriage, stillbirths, birth defects that occur in 7% of babies, which is 2 times more than is expected. In the Limpopo region, studies by our collaborators have shown that there's a very high level of urogenital malformation. So as many as 45 to 50% of the men in that area have urogenital malformations. There's an increased risk of cancer. Neurodevelopmental delays have been associated with exposure to organochlorines. And here we're looking at some work from Gina Muckle, where she was characterizing neurodevelopment in an Inuit child in northern Canada. Such tests have not been done in the Limpopo area, so we don't know if the same thing is occurring in that population is occurring in Canada North. And there's also a high risk of severe autism and intellectual impairment in these populations where you have high levels of DDT exposure. So we're wondering, could an altered sperm epigenome in response to DDT, could that be associated or implicated in the poor child health that can be observed in these regions where you have high levels of exposure? So as I mentioned, we had two populations. We had the Greenlandic Inuit population. In this population, there were 193 men that had been recruited for a previous study, the Innuendo population. And this is a study that was led by researchers in the University of Copenhagen. And we selected from their already collected samples, 47 semen samples that had normal semen analysis, that were homogeneous, relatively homogeneous for age, and had a range in DDE levels. And then in South Africa, we had 197 men volunteer from the study from sprayed and unsprayed villages. And from those 197 samples, we selected 50 samples that had normal semen analysis, again, were homogeneous for age and had a range in DDE levels. And we analyzed DNA methylation by methyl capture sequencing and histone modifications by ChIP-seq. We were only able to look at DNA methylation levels in the Greenlandic Inuit because the samples were limited in cell number. So here we're looking at serum levels of DDE, so the DDT metabolite, and we grouped the exposure levels to high, intermediate, and low levels. You can see here that the South African men, they had on average 12 times higher levels of serum DDE levels than the Greenlandic Inuit. So before I show you the results of the DNA methylation, I'm just going to explain to you, because you may not be familiar with the approach that we used. So this was an approach that was developed in our collaborators lab, the lab of Chiquetta Trasler. And what she had noticed in her studies is that in various models of exposure, what was found is that the regions that were most impacted tended to occur in these intergenic regions, so not at promoters. And this isn't that surprising with sperm, because sperm has a very unique DNA methylome in comparison to a somatic cell. So promoters, for example, tend to be hypomethylated in sperm, not all of them, but some. So she wanted to develop a technique where she could look at regions that she anticipated to be more sensitive to environmental exposures that currently aren't covered by the other techniques available at the time, which were the Infinium methylation array, the 850, and the EPIC. And so what she did is she profiled by whole genome bisulfate sequencing, so very deep sequencing, the same population of 30 men that we profiled, and chose regions to be captured by this custom capture by designing probes that not only covered the regions in the traditional assays, so in the EPIC array, but also covered 850, a million other CPGs that are in other regions, in particular in the intergenic spaces. So here we're doing that, looking at that comparison, and you can see that the intergenic space is most well covered in the sperm methyl capture in comparison to the other assays. And so she named these regions that are in the intergenic space dynamic CPGs, because she was able to show that these are the regions that are most responsive to environmental challenges in men. And she demonstrated that using a folate supplemented population of men. So what did we find in the sperm from men that were exposed to DDT? So here we're looking at the results from the Greenland population and the South Africa population. And so both showed differentially methylated CPGs, and there was a significant amount of overlap in those that were impacted in both populations. And that's important because that shows that there's consistency in regions in the sperm epigenome that are environmentally responsive to this toxicant. It's also important because these populations are genetically diverse and there are different routes of exposure. So the Inuit are exposed primarily through diet, and the South African men are exposed through direct exposure and things like inhalation. So there are regions that gained methylation and regions that lost methylation. And what we're looking here, these hatched marks, this is showing that the regions that were most changed as predicted occurred at these dynamic CPG sites that are localized in the intergenic regions. So what was really remarkable is that we found that in both populations, the gains and losses of DNA methylation was dose responsive. And so we believe this is the first time that any epigenetic mark in any tissue has been shown to be dose responsive to an environmental exposure. So we found that quite interesting. Next, we wanted to get some functional insight into what could be occurring in the genome. So we grouped the CPGs into what we call differentially methylated regions that are 500 base pairs apart. And you can see here on these graphs in our genome ontology analysis that the regions that gain, there are some located at the promoters, same with the regions that are lost in both populations. But again, there's lots of regions that lie in this intergenic space. When we did a GO analysis, we found in both populations that there was enrichment for processes that are involved specifically in nervous system development and also in reproduction. And then we wanted to look at, could the regions that are changed in the sperm be transmitted to the embryo? So we did this by identifying regions in the sperm that correspond to where you have DNA methylation at various stages through embryonic development. And we found that the dynamic DNA methylation regions were sperm to zygote persistent and sperm to inner cell mass persistent in both populations, suggesting that these marks could be a direct route to alter gene expression in the next generation. The other thing that I want to mention is that we also looked at where we found these changes. Did it occur in transposable elements? And indeed it did. And I mentioned just briefly before why we're interested in transposable elements, and that's because previous work, and it's summarized here in this schematic, has shown that in both the human and mouse, you have these minor and major waves of expression of transposable elements, and they can behave as enhancers, and they're really important for 3D genome organization. And certain classes of transposable elements, these have been shown to escape epigenetic reprogramming in the embryo. So this suggests to us that the marks that we're altering in the sperm or that are being altered by exposure could indeed be a route to disease in the next generation. Okay, then I'm going to briefly just show you our ChIP-seq data. So we looked at H3K4 trimethylation through ChIP sequencing, and shown here on the heat map are our findings, and we found that by differential analysis that there were regions that were changed that both gained and lost H3K4 trimethylation that was independent of BMI and age. And when we looked at the processes that are involved, just like we found for the DNA methylation, they tended to be involved in, for example, brain development and also in early embryo development. The next thing we wanted to know was, again, could this mark be transmitted to the embryo and does it persist? And sure enough, here we're looking at these scatter plots. We're looking at where you find H3K4 trimethylation regions that are altered at promoters in response to DDE across different embryonic stages in relation to gene expression indicated by the color. And sure enough, you can see that where the mark is found in the sperm corresponds to where you find the same mark in the embryo and corresponds to high levels of gene expression in the embryo. And then next thing we wanted to do was look at where we find this mark in relation to DNA methylation. And there was some overlap. There was a significant amount of overlap, but it was very minor. What was intriguing, though, is when we look at the functional processes where you find these two marks together, the most enriched, again, was nervous system development. So in conclusion, we've shown that there is an association between the DDE body burden and the profiles for H3K4 trimethylation and DNA methylation. That this also appears to be associated at the genes where we found changes that could be linked to, for example, the birth defects and the neurodevelopmental disorders and reproductive disorders that have been observed in these DDT-exposed regions. And we demonstrated that DDT exposure could be associated with generational effects as the marks that are changed persist in the embryo. Our results also show that it's really important to look for alternatives to DDT as a route for malaria prevention. And finally, summarizing all the work together, we think that there's enormous potential here for improving fertility in men and health outcomes of children by focusing on the links between the sperm epigenome and the environment and developing ways to palliate these effects and to prevent these effects. So in conclusion, I'd like to thank the members of my lab who have worked on these studies, as well as our funders, all the participants, our collaborators at the University of Pretoria and the University of Copenhagen, and our collaborators here in Canada, which have been essential for these studies, which involve many teams, not just my team. Janice Bailey's been involved, Dr. Chiquetta Trouser's been involved, researchers at Health Canada have been involved. Anyway, thank you. Thank you for your attention.
Thank you, Sarah. That was really impressive work, and I really appreciate the different lens you're giving to everything we're studying. It's great, and it's great to know that you're here in Quebec, so you can also be part of all these great minds and science that is grouped here to look at determinants of children's health. So you're really providing new data. We have a vast consultation now with all members of the network who are willing to contribute to that should be starting at 3:30. It's just an amazing talk that you gave. So if somebody has a pressing question, I think we could take one. Otherwise, I think we will jump to our participation. Yes, Marc-André, vas-y.
Une question difficile, Sarah. Do you think that part of the adapter to stress in the genome are specified at sensitive region, as you mentioned, and you can create hubs for those things to happen? So that's kind of a program adaptation. But the non-promoter close changes could be simply create diversity in the next generation gene expression so they can survive DDT in how many generations. So would have a combination of both specific response to stressor and randomized to create a diversity of gene expression.
So you're talking about maybe a compensatory response as well? Yeah, I think it's definitely possible, but trying to tease the two out would be very difficult. I think there's ways to do it, perhaps with some models of epigenome editing where you target areas that you hypothesize to be implicated in the maltransmission and areas that you expect to be implicated perhaps in a rescue. And so you could target one and then the other and then target both and see what happens and see if you do indeed get a rescue or a compensatory effect. So we're getting there to be able to kind of tease that apart by these large screening studies. And the tools are there now that we could do that.
Oui, d'ailleurs, Serge Maigrat fait ça, donc il n'est pas ici aujourd'hui, mais c'est quelqu'un aussi qui pourrait être dans le réseau.
Excellent. Oui, Gina, on ne peut pas résister de tes questions.
Hi, Gina.
Hello. Is it fine if I ask you a question in French, Sarah?
I'll do my best. Yeah.
OK. J'ai beaucoup apprécié ta présentation et ça m'amène toujours à réfléchir et à me dire que finalement, quand on ne considère pas la contribution du père, on sous-estime l'impact de ces contaminants ou encore même l'aspect protecteur des aliments, de l'alimentation. On a publié il n'y a pas si longtemps là, avec la cohorte MIREC des résultats qui montrent qu'il y a une association entre l'exposition prénatale aux phtalates et les traits autistiques chez les enfants, mais que cette association, elle est atténuée lorsque l'acide folique est élevé chez la mère en début de grossesse. Donc, il y a une protection offerte par l'acide folique. Cependant, les mamans et les papas, ils mangent à peu près la même chose, ou en tout cas, ils ont une convergence dans leur alimentation et Et l'acide folique, si elle est faible chez la mère au premier trimestre, il y a de fortes raisons qu'elle ait été aussi faible chez le papa au moment de la reproduction. Donc, il y a un gros pan de la réalité qui est absent dans nos études lorsqu'on ne considère pas le père, même au moment de la reproduction. Pour moi, la présentation que tu viens de faire et combinée à celle de Marc-André sont vraiment éloquentes. Merci.
Thank you. So I think you were commenting on the palliative effects of diet, and that you've seen them in pregnancy with women. And so there appear to be ways to reverse the damage in men. So I think there's a lot of, I agree that we should be looking at both mothers and fathers in all these studies going forward. But we have shown, and I didn't show the data today, in animal models that you can reverse the damage. So for example, you can feed a folate deficient diet, and then you can rescue the damage done to the sperm epigenome, as well as prevent the birth defects by feeding a folate supplemented diet. And then there's also been work done on the organochlorines by Janice Bailey, where her group used the rat model that was exposed to an organochlorine mix, and they were able to palliate some of the effects, again, by feeding folic acid. So there certainly is an ability, and there's a lot of hope in this, to prevent disease transmission. We just need to figure out, what are the appropriate doses, what are the appropriate times, and I think key is knowledge translation around this is one of the first things we can do, that dads matter too, and that their health matters in terms of having healthy children.
Okay, great. So thank you so much again. And everyone, now is the time vous êtes tous invités